The cell cycle in mitosis shows up in the biological and biochemical foundation section of the MCAT. You'll see them in standalone questions and in passages testing cancer biology or cell growth. The exam doesn't want memorized trivia. It wants you to understand how cells grow, how DNA replicate and divide, and how checkpoints keep the process under control. Let's break it down step by step so you can spot what matters on test day. The cell cycle framework. The cell cycle has four main stages: G1, S, G2, and M. G1, S, and G2 together form interphase, the growth and prep period. M phase is mitosis, when the cell divides its nucleus and cytoplasm. G1 phase, growth and function. In G1, the cell grows, makes proteins, and does its normal job. Before moving on, it checks, is the environment right and is the cell big enough? If not, the cycle pauses. S phase, DNA replication. Here the cell doubles its DNA. Each chromosome now has two identical sister chromatids joined at the centromere. This is crucial for mitosis because each daughter cell needs a complete genome. G2 phase, a double checking. During G2, the cell keeps growing and runs a quality check. Did DNA replication happen correctly? Any damage gets repaired before moving into division. Mitosis overview. Mitosis has four steps: prophase, metaphase, anaphase, and telophase. It ensures each daughter cell gets the same DNA. Prophase. Chromatin condenses into visible chromosomes. The nuclear envelope breaks down. Spindle fibers start forming. Metaphase. Chromosomes line up at the cell's equator, the metaphase plate. Each centromere attaches to spindle fibers, ready for separation. Anaphase. The spindle pulls sister chromatids apart, dragging them to opposite poles. Now each side of the cell has a full set of chromosomes. Telophase and cytokinesis. Nuclear envelopes reform. Chromosomes decondense back into chromatin. Cytokinesis overlaps, splitting the cytoplasm into two identical daughter cells. Checkpoints, the safety net. The cycle has three big checkpoints: G1 to S, G2 to M, and metaphase to anaphase. These act like brakes to prevent damaged DNA or incomplete division from moving forward. Regulation, cyclins and CDKs. Progression is controlled by cyclins and CDKs. Cyclins rise and fall with the cycle. CDKs activate only when bound to cyclins, phosphorylating proteins that push the cell forward. MCAT loves asking how changes in cyclin levels alter the cycle. Senescence and apoptosis. If conditions aren't right, cells may permanently exit the cycle. That's senescence. Or if they're damaged beyond repair, they undergo apoptosis, programmed cell death. Both processes protect the organism from harmful or cancerous cells. The MCAT doesn't just want you to label phases, it wants you to connect the dots. Know the sequence, the purpose of checkpoints, and how cyclins and CDKs regulate progression. That's how you'll score points when experimental passages bring up cell growth or cancer. With that said, here are just a few of our student success stories. Students who mastered these concepts, boosted their scores, and got into medical school. If you're studying for the MCAT, click the link below to get started with your own prep. We help students reach a 515 plus on the MCAT, and we'd love to do the same for you. And don't forget, subscribe to our channel for more MCAT breakdowns like this, and follow us on Instagram for extra tips and resources.